Three numbers on a blood panel stopped a research project cold, not because they were catastrophically wrong, but because they were impossible for the age of the men they belonged to. Vitamin D3, vitamin K2, and vitamin B12 were all reading at levels you would expect from men 30 years younger. The men in question were in their 80s, 90s, and past 100. They were shepherds in the Ogliastra highlands of Sardinia. None of them were taking supplements. None of them had ever heard of a longevity protocol. The study that captured those panels, the AKIA project, run by physician Giovanni Mario Pes and demographer Michel Poulain across 5 mountain villages starting in 1997, was not designed to find this. It was a longevity registry, a documentation of who was surviving past 100 at a rate with no parallel anywhere on the planet and what their lives looked like. The blood panels were incidental. The deviation they found was not. What I want to walk through is where those 3 numbers actually came from. Not a pharmacy, not a protocol, something harder to bottle than either of those. Hold on to the number. We are coming back to what it means. The AKIA study, Archivio Comunitario di Epidemiologia e Ancianos, which translates roughly as Community Archive of Epidemiology of the Elderly, enrolled over 2,500 individuals from the Ogliastra region. Pes and Poulain published the foundational analysis in 2004. It was not a nutrition study, it was a longevity registry, a systematic accounting of who was reaching extreme age and what their lives looked like. The vitamin data emerged not as a hypothesis, but as an observation. Here are the serum levels. Here is what they mean relative to reference ranges for aging populations. Here is the deviation. That deviation is what we are here to understand. The Ogliastra region sits in the eastern highlands of Sardinia, an island already set apart from mainland Europe by geography and by history. These are villages where the roads until recent decades were unpaved, where agriculture was subsistence-based, where the economy ran on sheep and goats, and what could be grown on the terraced hillsides. The cohort Pes and Poulain were studying had not been eating a longevity diet by design. They had been eating what was available, what their grandparents ate, what cost nothing because it came from the animals they kept in the fields around them. That context matters. The vitamin levels were not an achievement. They were a residue of a particular life. Before we go into the 3 values, I want you to understand why this matters for you specifically, not for a shepherd in Sardinia, but for someone reading a grocery store label or a lab result or a supplement bottle in 2026. Because the reason these numbers matter is not that the shepherds were doing something special, it is that they were not. The nutrients they had in abundance, most aging adults in the industrialized world are quietly running out of without symptoms obvious enough to catch until the deficiency is already doing damage. That is what makes the blood work from Ogliastra useful, not as a curiosity, but as a diagnostic mirror. Let's go through each one. Start with D3. Cholecalciferol, the form of vitamin D your skin synthesizes when ultraviolet B radiation hits it. There is a number attached to D3 sufficiency. In the clinical literature, 30 nung-link per milliliter is the floor for adequate serum 25-hydroxyvitamin D. Below 20, you are deficient. The average serum 25 OHD in elderly Italian mainlanders, people in their 70s and 80s, runs between 15 and 20 ng/ml. That is low. That is, in fact, the typical profile for an aging population in an industrialized country where most time is spent indoors. The Ogliastra shepherds were not indoors. An 87-year-old shepherd in Villagrande Stracili, not a name I can give you from a published registry, but a profile that appears in the AKIA observational records repeatedly, walks his flock between 10 and 14 kilometers a day across mountain terrain at between 400 and 800 meters of altitude. He starts before 7 in the morning. He is outside at elevation in thin air for 6 to 12 hours, 6 days a week most of the year. His skin makes D3 the way the body was designed to make it, through direct UVB exposure in the hours around solar noon, without sunscreen, without glass between him and the sky. The mechanism of what D3 does beyond bone health, and this surprised me when I first read through the immunological literature, involves a system called the innate immune response. D3 acts upstream of the inflammatory cascade. When a cell becomes infected or damaged, it triggers a signal that, If D3 is present in sufficient concentrations, modulates the intensity and duration of the inflammatory response. Think of D3 as the regulator on a furnace. Not the furnace itself, but the device that prevents the furnace from running at maximum heat when maximum heat is not required. Chronic low-grade inflammation, the kind associated with cardiovascular disease, cognitive decline, and accelerated tissue aging, is precisely the state that insufficient D3 fails to regulate. The inflammatory furnace runs hot. The body pays the cost. There is also a direct link to muscle tissue that most D3 coverage skips entirely. D3 receptors are present in skeletal muscle cells, not just in bone, not just in the immune system. The clinical literature connecting low D3 to sarcopenia, the progressive loss of muscle mass and strength that begins around 50 and accelerates past 70, is substantial enough that several geriatric protocols now treat D3 as a frontline intervention for fall prevention. Not calcium, not resistance bands D3. The shepherd in Villagrande is not losing muscle at the rate the clinical tables predict for his age. His D3 levels suggest one mechanism that may explain why. His muscles are still receiving the signal that tells them to maintain. And then there is calcium metabolism, the bridge to what we will look at next. D3 drives calcium absorption in the gut. Without adequate D3, the calcium in food moves through without being taken up. The body, needing calcium for ongoing cellular processes, begins pulling it from bone instead. This is the mechanism behind osteoporosis in elderly populations. It is not a calcium shortage in the diet. It is a failure of the transport system. What D3 does at sufficient levels is keep calcium moving in the right direction, absorbed from food available for bone maintenance, not scavenged from skeletal stores. The shepherd's skeleton at 87 shows the result. Dense, load-bearing, still climbing. There is one more dimension to D3 that the Sardinian data illuminated indirectly. And I want to be precise about what is established versus what is still being studied. The IKEA researchers observed that the centenarian cohort had lower rates of certain autoimmune markers compared to age-matched mainland Italian populations. D3 is known to modulate T-cell regulation, the part of the immune system that when it misfires attacks the body's own tissue. Whether the D3 levels in the Ogliastra shepherds were directly responsible for those autoimmune differences is not something the observational data can establish. The correlation is there. The mechanism is biologically plausible. The controlled trial that would settle the question has not been done at this age cohort. I mention it not to overstate the case, but because it is part of why immunologists have been paying attention to D3 far outside the bone health literature for the last 2 decades. Now move with me to the second piece. Vitamin K2, menaquinone, specifically the MK4 and MK7 forms, is a vitamin with a targeting problem at its core. And that problem is the center of why the Sardinian diet matters more than a simple list of foods would suggest. The targeting problem is calcium. Calcium is essential for bone density. It is also dangerous in the wrong locations, specifically arterial walls. When calcium deposits in arterial tissue, the vessel stiffens. Stiff arteries are less elastic, less able to absorb the pressure wave from each heartbeat, and they generate the conditions for cardiovascular events. This is not a minor concern. Arterial calcification is one of the most studied mechanisms in cardiovascular aging, and it is consistently associated with both heart disease and stroke risk in populations over 60. K2 is the postal system. The protein it activates, matrix GLA protein, known as MGP, is present in arterial walls specifically to bind calcium and redirect it toward bone. When K2 is absent or deficient, MGP stays inactivated. The calcium has no forwarding address, and it deposits in the arterial wall. When K2 is present, MGP is active. The calcium is rerouted. The distinction is not subtle. It is the difference between calcium building your skeleton and calcium hardening your arteries. When I first encountered the Rotterdam study data on this, Gelijns and colleagues published in 2004 examining K2 intake against coronary calcification in a cohort of almost 5,000 Dutch adults, the inverse correlation was steeper than I expected. Higher dietary K2 intake tracked with lower coronary calcification and separately with lower coronary heart disease mortality. The effect was dose-dependent. The association was specific to K2, not K1. K1 comes from green vegetables. K2 comes primarily from fermented foods and animal fats. And this is the distinction that almost never gets communicated clearly. Eating more leafy greens gives you K1, which has its own important functions in blood clotting, but it does not meaningfully raise tissue K2 levels. The arterial protection requires the fermented form. Here is where the Sardinian kitchen enters. Pecorino Sardo, Fiore Sardo. These are aged sheep cheeses, traditional raw milk made with natural rennet, aged between 6 and 18 months. The bacterial cultures in the fermentation process convert K1 from the sheep's grass-fed diet into MK4 and MK7, the two K2 forms with the longest residence time in arterial tissue. This conversion does not happen in pasteurized cheese. It does not happen in fresh cheese. It happens in the aging process, in the months of microbial activity that transform raw milk into something structurally different from what it started as. The Ogliastra diet is not a vegan diet and never was. It centers on legumes, sourdough bread made from durum wheat, seasonal vegetables, and wild greens, but it includes substantial amounts of aged sheep cheese from animals that graze on the very highland pastures the shepherds walk through. The cheese is not a treat. It is a meal component daily across decades. A 96-year-old shepherd in Predastafogu, in a profile described across the IKEA documentation, eats pecorino at most meals. Not a wedge, a real amount. He has done so for most of his adult life. He has no known arterial calcification event in his medical record. That is one data point, not a controlled trial. But the biochemistry behind it traces directly to what Galijnse measured in Rotterdam. There is a note I want to add here that the study does not settle the optimal daily amount of K2, and whether the MK4 and MK7 forms have equivalent effects is still an active area of research. The Rotterdam data is observational. There is no randomized trial establishing the exact dose. What the data establishes is the direction of the relationship. The Sardinians got there through food, not through calculation. I also want to address something that comes up every time I discuss K2 in a longevity context because I hear it constantly in the comments. The question is, what about K2 supplements? If the cheese is doing this through bacterial conversion, can a K2 capsule replicate it? The honest answer is partially, and the gaps matter. The supplements that contain MK7 are generally well absorbed. The pharmacokinetic data on MK7 supplementation shows it reaches arterial tissue, but what the supplement does not replicate is the food matrix. The combination of fats, proteins, and cofactors present in aged dairy that influence how K2 is absorbed and distributed. Whether that matrix effect is clinically significant is still being studied. It is not settled. What is settled is that the K2 in traditionally fermented aged cheese is bioavailable, that the Rotterdam cohort was getting their K2 from food rather than supplements, and that their arterial outcomes were measurably better for it. I am not telling you to avoid K2 supplements. I am telling you that replacing the cheese with a capsule and assuming you have replicated the mechanism is a simplification the evidence does not fully support. From the food, we go to the third panel in the blood work. B12, cobalamin, is the vitamin that aging populations are most reliably losing. The mechanism is straightforward, and once you understand it, you realize why the conventional solution of eating more B12-rich foods often fails for people over 60. B12 absorption depends on a protein called intrinsic factor produced by cells in the stomach lining called parietal cells. As those cells age and the stomach becomes less acidic, intrinsic factor production decreases. The B12 in your food is present. The transport system that gets it through the intestinal wall is impaired. Serum B12 falls slowly over years. The symptoms, numbness, fatigue, cognitive fog, balance problems, look like aging. Many clinicians miss it precisely because it looks like aging. It has been, in fact, looked like aging in millions of people for decades while being something diagnosable and addressable. There is a second layer that connects B12 to something most people do not associate with it. Brain volume. B12 is directly involved in the clearance of homocysteine, an amino acid that accumulates when B12, along with folate and B6, is insufficient. Elevated homocysteine is associated with accelerated cognitive decline. The studies on this are not peripheral. The Oxford B vitamin trial and subsequent meta-analyses have shown that reducing homocysteine with B vitamin supplementation slows brain atrophy in individuals already showing signs of mild cognitive impairment. The effect was concentrated in the hippocampus, the region associated with memory consolidation, and one of the first areas to show deterioration in early Alzheimer's pathology. This is not a marginal finding. It is one of the cleaner intervention signals in the cognitive aging literature, and B12 is at the center of it. The fully plant-based elderly population faces the highest B12 risk. There is simply no reliable plant source of B12. Certain algae contain B12 analogs that do not function as true cobalamin in the human body. Nutritional yeast, unless specifically fortified, does not reliably supply B12. The fermentation myth, the idea that traditionally fermented foods supply B12, is not supported by the evidence in any meaningful therapeutic quantity. The Sardinian peasant diet was largely plant-anchored, but never fully plant-based. The small animal protein component, a weekly lamb or mutton stew, slow-cooked on the bone, eggs from free-range hens, sheep cheese, occasional offal in traditional preparations, was enough. Not a large amount, enough. And crucially, it was enough because it was delivered in small doses across multiple days, rather than in a single large weekly serving that the aging stomach cannot process efficiently. A 102-year-old woman in Perdastafogu, in the village records referenced in the IKEA documentation, still keeps 3 hens. She eats 1 or 2 eggs several times a week. She eats a small plate of the local cheese daily. She eats meat perhaps once a week, sometimes less. Her B12 levels from the late-life blood work captured in the study were within normal range. Normal range for a centenarian is not typical. Most centenarians in the industrialized world do not have B12 within normal range without supplementation. I want to reframe that before I continue. When I say the 102-year-old woman's B12 was within normal range, I do not mean it was normal the way a 30-year-old's would be. The reference range in geriatric medicine is itself a moving target. And the debate about what constitutes optimal B12 versus merely sufficient B12 for neurological protection is ongoing. What the IKEA data captured was the relative comparison. Her levels versus age-matched populations in other regions. She was above the cohort average, not necessarily above a young adult's level. The distinction matters if you are trying to interpret your own lab results. A B12 reading of 220 pg/ml may technically be within range on a standard panel while still being too low for optimal neurological function in someone past 70. The floor matters. The floor the Ogliastra centenarians were above was a higher floor than the one most laboratories use. Here is what the 3 values taken together reveal. This is not a video about which supplements to buy, and I want to be direct about that, not as a disclaimer, but as the actual finding. When researchers analyzed why the Ogliastra vitamin levels looked the way they did, the answer was not a pharmacy protocol. It was a pattern of sun, food, fermentation, and animal protein frequency that, when taken together, consistently supplied 3 nutrients that aging bodies otherwise run short of. The shepherd gets D3 from 12 hours of daily outdoor movement at altitude, not a D3 capsule. The cheesemaker gets K2 from 6 months of bacterial fermentation in a wheel of raw milk pecorino. Not a K2 supplement. The grandmother's hens and her weekly stew supply B12 at a dose that aging absorption can still handle. Not a methylcobalamin tablet. There is a point in the Acciaroli research where Pes and Poulin tried to isolate which lifestyle variable was doing the most work. The honest answer from the data is that the variables are not separable. The outdoor activity that generates D3 is the same physical labor that builds the bone density that K2 is protecting. The fermented dairy that delivers K2 is the same food that provides B12. The social meal structure that produces the once-a-week lamb stew is the same structure that prevents the isolation associated with cognitive decline. You cannot pull one thread without pulling the others. The study was not designed to generate a list of interventions. It was designed to document a life. What it documented, incidentally, was a life that had solved 3 of the most common nutrient deficits in aging populations without ever trying to solve them. Write this down. We are coming back to it. The paper that is hardest to translate for an American kitchen is not the Rotterdam study or the Icaria longevity registry. It is the observation that runs underneath both of them. The nutrient profile of the Ogliastra centenarians is not the result of optimization. It is the result of a pattern of living that happened to produce optimal results. The shepherds were not tracking their serum 25 OHD. They were walking their flocks. The grandmothers were not calculating K2 from MK4 and MK7 sources. They were making the same cheese their mothers made. The hens in the courtyard were not a B12 delivery system. They were 3 hens in a courtyard, and they laid eggs, and the eggs were eaten. That is important because the wellness industry translates these findings into product lists, and in doing so, loses the mechanism. The mechanism is not the cheese. The mechanism is decades of a particular relationship with food, animals, sunlight, and physical terrain, producing a biochemical profile that happens to protect the brain, the skeleton, and the arterial wall at the same time. You can approximate the outputs, aged cheese, time outdoors, eggs several times a week, without approximating the life. And approximating the outputs is still worth doing. The Rotterdam data does not require you to be a Sardinian shepherd. It requires you to get K2 into your diet through food that has actually been fermented. That is replicable. What I am cautioning against is assuming that once you have the list, you have the answer. What changes in your week? 3 things practically. Not a protocol with 12 steps. 3 things. The first is outdoor time in direct sun, without glass, before noon. 20 to 30 minutes of skin exposure is enough to move the serum D3 needle in most adults. The exposure needs to be to actual skin. Forearms, lower legs, the back of the neck. A window does not count. UVB does not pass through glass. The reason the shepherds were synthesizing D3 at the rate they were is that nothing stood between their skin and the sky for 6 to 12 hours a day. If you are past 60, dermal synthesis of D3 is slower. The skin's conversion efficiency drops with age. Plan for closer to 40 minutes for the same effect. The shepherds got 12 hours, you need 40 minutes. That gap is real, which is why D3 supplementation is one of the few cases where the clinical literature supports supplementation across the board for adults over 65. The food and sun pathway works, but the window for adequate synthesis narrows with age, and in many latitudes and seasons, it closes entirely. If you live above the 37th parallel, roughly the latitude of San Francisco or Washington, D.C., from late October through March, UVB is too weak for synthesis, regardless of how much time you spend outdoors. You can be outside for an hour at noon in January in Chicago and synthesize essentially nothing. This is a geographic fact, not a personal failure. And it is why serum D3 deficiency is dramatically more common in northern populations during winter months. Supplementation during those months is not optional for people in this latitude band. It is a replacement for a biological process the sun is no longer performing. The second is fermented dairy. Not processed cheese, the distinction matters. Processed cheese, the sliced, wrapped, shelf-stable kind, has been heat-treated in ways that destroy the bacterial cultures responsible for K2 conversion. Traditional aged cheese, Parmesan, Gruyere, aged cheddar, Gouda aged 12 months or more, carries K2 in meaningful amounts because the bacterial activity during aging has had time and temperature to do its work. Pecorino Romano is the closest widely available equivalent to what the Ogliastra shepherds ate. Gruyere aged 18 months is another reliable source. Natto, if you can find it, carries more MK7 than any dairy product. 1 to 2 Oz of traditionally aged cheese daily is a range that aligns with the Sardinian intake pattern in the IKEA documentation. Not at every meal. Daily. The third is B12 frequency, not B12 dose. This is the one most people get wrong, and the misunderstanding comes from treating B12 like a nutrient you can stockpile. You cannot, at least not efficiently, past a certain age. The absorption problem in aging is not improved by megadosing because the problem is not the amount of B12 you consume. It is the amount your stomach can transport per meal. Intrinsic factor saturates at about 1.5 micrograms per sitting. A 1,000 microgram B12 supplement delivers roughly 1.5 micrograms of that into your bloodstream through the intrinsic factor pathway. The rest passes through or is absorbed through a separate, much less efficient, passive diffusion pathway. What works is frequency. 2 eggs in the morning, a small amount of animal protein at a later meal, or dairy, not a massive weekly steak. Daily modest amounts reliably. The grandmother in Père de Fogue did not eat a large piece of meat once a week to compensate. She ate a small plate of cheese every day, a couple of eggs several times a week, and a modest serving of meat when it was available. Her stomach processed each of those servings through the intrinsic factor pathway as designed. Even though that pathway was running at reduced capacity, frequency compensated for the reduced rate. That is the lesson. Not more at once, more often in smaller amounts. One more thing the blood work showed. The K2 levels in the Ogliastra cohort were higher than Galines's Rotterdam data would predict from the amount of aged cheese alone. The working hypothesis among the researchers is that the traditional Sardinian sourdough bread, made with wild fermented cultures from grains grown in the same highland ecology, may carry bacterial K2 precursors not typically present in commercial bread. Certain strains of lactobacillus involved in sourdough fermentation have been shown in laboratory conditions to produce menaquinones. Whether the amounts produced in traditional sourdough loaf are therapeutically significant is not something the literature has established. The fermentation research on sourdough K2 is thin. I'm not going to tell you your sourdough is delivering K2 in meaningful amounts because the data does not yet say that. But I notice it, and I mention it precisely because I think the honest answer is the story is not complete. The AKEA data pointed to an unexplained surplus in the K2 readings that the cheese alone does not account for. The sourdough hypothesis is where the researchers are looking. We do not have the answer yet. What we do have is the pattern, and the pattern taken across D3, K2, and B12 tells a coherent story about what happens to a body when it spends decades outdoors eating food that has been through real microbial processes and maintaining an animal protein intake modest enough that aging absorption mechanisms can keep up with it. The Ogliastra shepherds did not live long because they were optimizing. They lived long because a particular configuration of life, terrain, food, sunlight, animals was producing the biochemical conditions that most of us are trying to engineer from a supplement aisle. The replication is imperfect. The principles behind it are not complicated. A last image. The aging rack in a traditional Ogliastra cantina, wheels of Fiore Sardo stacked in cool darkness, rind thickening over 6 months of bacterial work is doing something that modern industrial cheese production does not replicate. The microbiology of that process, the conversion from K1 to MK7, is happening in the dark, quietly, the way biological processes mostly do, without anyone tracking it. The light through the stable window in the morning, where the hens move across the stone floor before the shepherd opens the door to the mountain. The bowl of minestrone on the kitchen counter with the pecorino crumbled into it before it cools. The walk up the hill and back 6 days a week, sun on skin for 87 years. The blood work values did not come from nowhere. They came from there. One more thing before you go. The 3 practical adjustments I gave you, outdoor time, traditionally fermented aged cheese, B12 through frequency, are not a protocol designed for Sardinia. They are the minimum viable translation of what the AKEA data showed into a life that is not lived in a highland village. They are not perfect. They are replicable. And the difference between someone who makes those 3 adjustments and someone who does not, compounded across the next decade, is the difference between the blood work of a 90-year-old in Ogliastra and the blood work of an aging population that never got the memo. You have the memo now. If this exploration of the Sardinian blood panels has changed the way you look at your own health and the simple habits of your day, please subscribe and like this video. We are dedicated to uncovering the historical and biological truths that have been overlooked in our modern world. Let's plant the seeds for a longer, stronger future together. The next chapter of human longevity will be open soon.